Hello everyone, my name is Subhas and I'm going to briefly present my work today on morphine-induced immunosuppression is mediated by microRNA 155. So for introduction, here we know that we are currently going through a major opioid epidemic crisis, and at the basis of this is a chronic morphine use and abuse, which severely compromises the immune system, leading to different, many different opportunistic infections. And therefore, we are interested in finding out how the OPRM1 gene, which is a mu-opioid receptor gene, is regulated by different cis-acting elements. When it comes down to cis-acting elements, we are trying to find out how transcription factors like PU1, which is actually behaving as a silencer complex, is mediating this OPRM1 gene regulation. So in this study, we are trying to find out epigenetic regulation of OPRM1 through microRNA 155 in LPS-induced immune activation. And therefore, here you can see on the cartoon that we have a distal promoter. Here is a distal promoter regulatory sequences, and we have a proximal promoter. And this here is a P1 binding site, which is working as behaving as a silencer complex, leading to negative regulation of OPRM1 gene. For methods, we used mice for in vitro experiments and peritoneal macrophages for in vivo experiments. And in both the cases, we treated them with morphine for 24 hours, followed by the LPS treatment for 6 hours. For results, the first slide here is talking about the microRNA 155 expression through microarray experiment. So as you can see here, that LPS upregulates the microRNA 155 expression in wild-type rats, but also does in more knockout mice. But what is surprising here is that morphine brings that LPS-induced activation of microRNA 155 to control levels, almost to control levels, but in more knockout rat, it does not happen. That simply means that microRNA 155, among other microRNAs, are mediating this role. So we are trying to study this microRNA 155 in this case. We wanted to locate where this microRNA is showing its activity. So we showed that in a spleen, LPS drives this microRNA 155 expression, but this is not seen in bone marrow or the thymus cells. We further confirmed that in the spleen, we have a splenic macrophages, which is showing the LPS-induced activation of microRNA 155, which was not seen in the T cells or in the more knockout mice. So we are targeting the splenic macrophages. So for all our further experiment, we used the cell line, which is J774.1, which is a peritoneal macrophages. So first thing first, we wanted to confirm whether LPS drives the mu-opioid receptor upregulation or not. It does. We confirmed it by qualitative and quantitative experiments. And we also confirmed that microRNA 155 is upregulated in the LPS treatment, and morphine brings it down. So our further experiment, we wanted to see how this microRNA 155 is regulating the OPRM1 gene. So we confirmed what are the target proteins. Among other, we found it out that PU1, which is a transcription factor, is one of the target proteins for microRNA 155. So you can see here that LPS brings it down, the PU1 expression, while morphine pretreatment rescues that effect. And this was further confirmed by the PU1 quantitative PCR. So in conclusion, we can conclude that LPS treatment drives this microRNA 155 expression, and morphine treatment reduces this LPS-induced microRNA 155 expression in the splenic macrophages as well as the macrophage cell line. Also, we showed that microRNA 155 targeted transcription factor, PU1, which is a silencing element, and thus negatively regulates the OPRM1 gene expression. So we concluded that OPRM1 gene is regulated through epigenetic regulation by microRNA 155 mediated through PU1. So for future directions, we are trying to find out how this is happening. So we are going to do the protein-DNA interaction by EMSA experiments and chip assay to confirm the PU1 binding site to the negative silencing element, as well as interaction between the microRNA 155 to PU1 further. With that, we would like to thank the CHS research organizers for letting us present our work. Thank you very much.